We are a collaborative, interdisciplinary lab, a feminist research lab that works to generate concepts, methods, and theories for scientific research on sex and gender. We are working on advancing the intersectional study of gender in the biomedical and allied sciences. We work to counter bias and hype in sex difference science and to enhance public discourse surrounding the sciences of sex and gender. So we're interested in these sorts of questions. How do we advance work to understand apparent sex differences while counteracting sex essentialist assumptions? What resources can we offer conceptually to researchers and to policymakers seeking to better understand gender and sex? And can we shift the conversation around sex, gender, and sex differences to be more transdisciplinary, more intersectional, and more accountable? So to give a concrete example of this work, in the spring of 2020, at the start of the COVID-19 pandemic, our lab members were kind of thrown into the virtual environment, as we all were, and many of us found ourselves, frantically grappling with a lack of childcare, and switching to teaching and learning online, and we were hyper-focused on COVID, as everyone was, and we started to notice a rising discourse of biological claims about variations. There were claims about the biology of race, there were claims about the biology of sex, and people turned immediately to biological factors to explain these variations, looking, for example, in the case of sex, at sex hormones, at genes, at immune factors, and so on. So to give an example of the kind of rich critical work that we do in our interdisciplinary lab, this was an early, rushed to publication Nature piece in 2020 that argued that it was biological immune differences between males and females that was causing the apparent sex disparity in some locations at the beginning of the pandemic. Men were dying at a rate of almost 2 to 1 compared to women. And this article argued for different vaccine approaches and therapeutic approaches, 2 different pathways, one for men and one for women. Well, they argued, for example, that the immune landscape in COVID-19 patients is considerably different between the sexes, and it was widely disseminated. So we have a very binary sex difference claim. We scrupulously reanalyzed the data, and we demonstrated that there were many fallacies in this study, including that myriad analyses were conducted, but actually very few sex differences found. The sex differences swam in and out of view depending on the analysis done, and that the data set was too limited to support the strong conclusions of the paper. To give an example, when adjusted for age and BMI, no statistically significant sex differences replicated across the cohorts considered in the study. To us, this is just every day. It's indexical of the huge problems in sex difference analyses. If you want low-hanging fruit for teaching or analyzing fallacious reasoning in difference claims, go to the sex difference literature. So we were able to publish this paper that was a reframing. Remember, there's always the question of similarities. How do we show similarities? Is that something we can do with data? We showed that their data actually demonstrated a very similar immune response to COVID-19 across the sexes. Also, to better understand COVID-19 claims and data, we started our own data tracker. We were the only data tracker in the world to collect longitudinal, geographically distributed data, which we did every Monday morning for 18 plus months across the United States. And so we created this publicly accessible data tracker, still available for free use, and many visual representations and contextualizations and charts and figures on our website. And even in the early days of the pandemic, it was quite clear that this apparent sex disparity varied greatly across geographic location. It was far from straightforward. It varied across location, but also across time. So for an example, in New York State, in the very early stage of the pandemic, you had that 2-to-1 ratio, that striking sex disparity. But by the time that the public health regulations and masking came in and the lockdowns came in, by May, it was about 1-to-1. So, and for the rest of the pandemic, it remained about that close. So cumulatively, it still looks like there's a huge sex disparity out of New York, but actually that was very time slice dependent. Next door to New York, in the state of Connecticut, they have had almost equal parity in the sex disparity in COVID-19 outcomes. So right next door, totally different outcomes. Some of our key findings came from intersectional analyses that we did. We were the first to do race by sex analyses. So how do sex disparities, we asked, vary across groups defined by race and ethnicity? This is as much a story of the dearth of available data for doing these kinds of intersectional analyses as anything, but in two, I think, really revealing states, Michigan and Georgia, we had the data to do these analyses. And we didn't know what we would find, but our results were stark. We did find that Black men had higher mortality rates than any other race or sex group, revealing a particularly vulnerable subgroup. But contrary to the story of men as uniquely vulnerable, Black women had 3 to 4 times the mortality of white men or Asian Pacific Islander men. And we also found that the sex disparity in mortality varied widely between different groups. That is, the mortality difference between Black men and Black women was much larger than the corresponding disparity between white men and white women. Okay, so to just put a point on it, here are some of the key findings. Black women had COVID-19 mortality rates in our data set that were 4 times higher than that of white men. Black men had higher rates of mortality than any other sex and racial group, over 6 times higher than the rate among white men. The disparity in mortality rates between Black women and white women is over 3 times the disparity between white men and white women, and the disparity between Black men and Black women is larger than the disparity between white men and white women. Okay, so I emphasize this and dwell on this because this is exactly the kind of thing you miss if you only do male-female sex comparisons, right? It's just a classic example. So what did we find overall from our tracker? I realize this is really small on your screen, but in short, we found that throughout the pandemic, the sex difference in case rate and in mortality rate varied widely. Usually women had higher case rates than men, and we published a paper on the difference in denominator between women and men for determining case fatality rates that has not been noted anywhere else. So higher case rates among women will artificially deflate their mortality rate. Many states resembled Louisiana, with sort of women having slightly higher case rates throughout the pandemic, but there were states with little difference in case rates, and then there was Texas, where men had much higher case rates. Mortality rate. The mortality rate was nearly always slightly higher in men, which is true in general and was true prior to the pandemic. Mortality rates for men are higher. But in some states, men had starkly higher mortality right from the beginning, and it persisted, whereas in others, like I mentioned with New York, there was change throughout the pandemic where there was more of a closing in that disparity. And here's a big, messy figure that I'm actually kind of proud of, but it's very, very busy, but overall it shows that men had higher mortality than women, that is right above 1. But there's significant variation. So for example, in the state of Connecticut, for 20 weeks of the time that we tracked, women had higher mortality than did men. So this is, and the gray lines equal all the states. So considerable variation in heterogeneity. And I emphasize this because one of the simple practices that we can use to explode essentialist categories is to bring to the fore context and variation. It's a simple practice, but a powerful one, and one that we try to model in the work that we do. So in short, we found that this gender-sex disparity, thought of as resulting from a biological sex binary rooted in innate biology, actually varies by context. It varies across social groups, it varies over time, and it varied between place. So for us, this opens up a conversation about social factors linked to COVID-19 gender-sex disparities, including health behaviors, I think most strikingly occupational patterns, which are strongly gender stratified, and pre-existing health disparities among and between men and women. And as we demonstrate, you just can't do this work without intersectional analysis. So we are able to argue the importance of context, the context sensitivity of these patterns, in other words. In sum, our work is oriented toward constructing an argument that a measured outcome, a quantitative, biologically measured outcome of a difference, does not entail a sex-based mechanism. We good? Gender-related social and contextual factors need to be considered when faced with a disparity associated with sex, and most importantly, gender is intersectional and multi-level. A unidimensional analysis will obscure important variation. Yeah?
Can you just clarify what you mean by multi-level?
So let's see. That's probably a precise term in your world that is a little different from me. So we talk about gender being enforced at multiple levels, at the social institutional level, at the level of interactions, at the interactional level, and at the level of individual ideology. That's itself a distillation of great complexity, but this is levels of analysis that make it a fundamentally relational and multi-layered construct. Sometimes gender at one of those levels can modify the effect of gender relations at another level, for instance. Thank you. Okay, so what's this business about sex as a biological variable? Okay, this is sex as a biological variable, or SABV, is a major question right now for funding agencies, for journals, for pharmaceutical companies, and this leading conceptualization of sex in biological research argues for the consideration of sex differences at every level of biological analysis. The real-life implications are, as I will argue, to produce and proliferate binary sex comparisons across biomedical research. So here's the context. The policy was introduced in 2014, requiring researchers to include analyses of male-female comparisons, including in cells and tissues and animals used in all funding applications. This is not the same as an earlier policy put in place in the early 90s, which required the inclusion of embodied, whole women in clinical trials. This is preclinical research in the experimental bench laboratory context. And it argued for doing so on the basis of redressing men's and women's health disparities. So the idea is the cleanest route to addressing sex disparities in health outcomes is doing more sex difference research in the lab. This is part of a large institutional effort led by women's health advocates and by elite sex difference researchers to advocate for the study of biological sex differences in preclinical models. And it followed those earlier successes of requiring inclusion in clinical trials. And it was constructed by these policy elites through an impressive strategy of building new journals, new awards, new conferences, the Organization for the Study of Sex Differences, the journal Biology of Sex Differences, a really concerted campaign to create a knowledge platform for sex difference research over the last 15, 20, 25 years. Now, sociologist Steve Epstein has written about this dynamic in biomedicine, and he calls it the inclusion and difference paradigm. There have been a half-century of efforts by various groups to be included in our publicly funded scientific research that is supposed to serve everyone. And this practice, he argues, of bargaining for inclusion of a group of research subjects in medical research advances often by claiming the possibility or probability of medically relevant biological differences. So it's in this dance with concepts of liberation, social justice, and equity, as well as with concepts of essential biological difference. And this headline, I'll read it to you. I think one of the calls of this gathering was around concepts of justice and fairness. And these are active in these literatures. Female mice liberated for inclusion in neuroscience and biomedical research, read one headline around the time of implementation. So, these calls for inclusion certainly are accompanied by strong binary essentialist claims, sort of womb-to-tomb ideas, head-to-toe ideas of essential, fundamental sex differences. And there's been a debate around these policies. And the two central questions, I'm going to move real quickly through this, the two central questions, and I've been one of the leading critics of these policies, are one, is this true? If we study sex differences more in these preclinical materials, is it actually true that this will facilitate health justice? And I've argued no, there's actually no example of that, and it's likely, possibly, to proliferate problematic claims. And then the second question, which will be more my focus today, is this thing called sex actually conceptually coherent as a biological variable? Does sex cause things? Is sex a variable in preclinical research? I have argued that it's so complex, so contextual, that requiring its study in this decontextualized way across all of these areas of biomedicine is likely to produce conceptual muddle. So those are the two major areas of debate. So if we think about this literature, again, it has created a discourse, I call it SABV discourse, where sex is everywhere. This headline says, I don't know the question, but sex is definitely the answer. And that's kind of the mode here. One of the mantras, if you go to just about any women's health institute, is every cell has a sex, either male or female, right? And so it's this omnipresent idea. It is that has gathered a lot of energy and excitement around it, right? And I call these the two hypotheses, the hypothesis of difference, so there's difference throughout, and the hypothesis of omnipresence. So Janine Clayton of the NIH Office of Research on Women's Health is fond of saying every cell has a sex, each cell is either male or female, right? And in this vision, sex is a ubiquitous element, not localized to gross regions of sexual dimorphism. And this is a sex essentialist approach to the construct of sex. It argues that there's so much difference that studies done in one sex may not be generalizable to studies done in the other, and it's omnipresent. That is, all biological systems and materials have a sexual identity, right? There's an essence, male or female, and so hence comes the inclusion prescription. So, and here, for example, is a guidance given to reviewers of NIH grants, and this is probably hard to see from where you are, but what you need to know is that the only thing that reviewers need to be attuned to is the ability to test for sex differences, yes or no, by which they mean comparing male and female types, disaggregating by male and female. So I call this SABV discourse. That is, there's a number of assumptions embedded in this, that it's presented as just good science, but there's more to it. There's a politics, there's a set of values, and this includes what I see as the following. First, that studying sex differences is apolitical, has no implications for the construct of gender. Sex is biological, gender is cultural, sex is material, it just can be objectively measured. Sex can be studied independently of gender, gender meaning the set of cultural expectations and scripts, roles, behavior associated with masculinity and femininity, in contrast to sex, which generally refers to the biological material components of male-female difference. And then secondly, at the same time, while saying it's apolitical, they also argue that studying sex differences fulfills the mandate of gender equity. So it's the analysis of biological sex differences is positioned as fundamental and central to women's health equity. And the operationalization of this is the inclusion of equal numbers of male and female specimens in scientific research. And that's posed as a gender justice issue. And third, studying sex difference is just good science. Just good science. Sex and gender research is a tool often in this discourse for innovation, for discovery, for precision optimization in basic science and medical research. I feel a sadness at the embrace in this discourse of what I see as superficial and essentialist constructs of sex and gender and the distinction between the two, and of gender justice, really, that is present in this SABV discourse. As I'll hopefully show you in a variety of ways, there's strong evidence that none of these things are uncontestably true. For example, a few years ago, when the U.S. Health and Human Services Administration issued a memo arguing that sex means a person's status as male or female based on immutable biological traits identified by or before birth, and arguing that this definition is necessary for ascertaining a person's sex on a biological basis that is clear, grounded in science, objective, and administrable. That memo cited SABV policy and documentation. If you're interested more in that policy terrain of these debates, why it matters in the world, we have a recent piece, Law, Policy, Biology, and Sex in Science, that puts together our reflections and suggestions around this issue and tries to raise awareness of how these scientific concepts do not stop at the laboratory door and have implications for law and policy decision-making, which often relies on scientific authority, as you all know so well. Okay, so on to operationalizing sex in biomedical research. I want to give just one example of why sex isn't as coherent as a biological variable as it is presented in this literature. So you may know the example of the HeLa cell line, which was infamously derived in 1951 without consent from the tumor of Henrietta Lacks, a Baltimore woman, and it was cultured and immortalized to provide standardized research material for scientists, generating an astonishing 74,000 scientific publications. It actually became the most widely used cell line in 20th century biomedical research, particularly in critical areas like cancer research. Now, it's interesting on so many levels. Not only has the HeLa cell line been proven to contain also now the genomes of several viruses, but also the genomes of several of the researchers who have interacted with it. So much so that someone, I think perhaps rightly, suggested that it be given its own species name. It's unclear whether it is an issue of gender justice that so much cancer research has been done on this, so to say, female cell line, since cell lines themselves are such funky, odd objects that explode our concepts of categories. But I would argue that I use this example as just a way in to the hybridities and artifice of so many laboratory materials, suggesting that they have a sex that transfers to sex as we understand it in our social worlds seems to badly misunderstand the context specificity of these materials. Another example with cell lines, so-called male cell lines derived originally from a male organism. At least 40% of those cell lines have been shown to have lost their Y chromosome, which would be the critical genetic feature that would distinguish it from any other cell line. Similarly, when we work with cells, they are in a growth medium that contains phenol red, a weak estrogen mimic, and fetal bovine serum, which contains a mix of pregnancy hormones from both male and female fetuses. So the construct of sex, when we're talking about cell and tissue research, is an incredibly complex one. To summarize the critique here, when we're thinking about sex in the world of laboratory-based preclinical research, it just isn't true that male-female comparisons are always the best operationalization of sex. Many operationalizations of male and female are actually limited in their generalizability to all males and to all females. Claims about male-female sex difference in a model system are limited in their generalizability to embodied humans, and biological material does not always have a clear, stable, or functionally important sexual identity. And the final point is too complex for me to elaborate here, but not all differences between sexes are appropriately called sex differences. All of this is summarized in a paper called Sex Contextualism, and there's a teaching slide deck and explainer and handout for researchers available on the Gender SciLab website if you're interested in exploring that. I basically argue for a contextual view of sex. What sex is pragmatically and plurally defined in biomedical research. And what that does to our questions about fairness, inclusion, justice, is it puts the responsibility on the researcher. If sex is pragmatically defined, contextually defined, plural in biomedical research, and it's sensitive to its social context and implications, then researchers need to justify their choices in how they operationalize or attend to or not sex in their research. All right, over to sex, gender, and precision medicine. So this is a recent piece that somehow you noticed, Cynthia, where I'm starting to think about precision medicine and big data and what it all means from a gender and sex perspective. So I'm so interested in the conversation around this. So a few thoughts. Precision medicine initiatives will define the landscape of science funding and medical practice for decades to come. So in this work, a question is, Where is sex and gender? Can we build a precision medicine, whatever that is, that attends to, interrogates essentialisms of all kind, and cultivates and affirms nuance and complexity, and is accountable and responsible in the way it uses sex and gender? Or are we going to get sex as a biological variable, binary thinking, check the box by including a sex comparison, and thereby reifying this kind of construct? It's an open question right now. Here is a figure from a recent Cell article outlining the future of precision medicine. It was lead authored by All of Us director Joshua Denny with Francis Collins. And it demonstrates that things like exposomics, phenomics, and related big data efforts to map human behaviors and environments are currently envisioned as the pillar of the future of precision medicine, alongside, of course, lots and lots of genomic data, right? So at the heart of it, really, and that's where all the money has gone so far, is genome sequencing. Global genome sequencing, routine use of clinical genomic data. But what this figure sort of represents is that there is a vision of a robustly socially situated and culturally sensitive future for precision medicine. So while we underscore that genomics is pretty much what they've done so far and is the bulk of the investment, there is this vision that has social and environmental approaches, at least at the margins, and they're underdeveloped right now relative to the genomic technology, but in this vision, they are on an equal plane with them. Here's another such image. Exposomics is another one of these post-genomic precision, big data sciences that argues for bringing in quantitative sophistication to our operationalizations of environmental factors. So in other words, meeting the data richness of genomes with data richness representing the environment. So Jeffrey Miller envisions exposome-based EWAS to be conducted on the scale of GWAS, and the systematic identification of the impact of non-genetic factors and chemical exposures that would enable the establishment of exposome risk scores akin to polygenic risk scores. Now, there's a space here for contestation. On the one hand, there is some promise. In that Denny and Collins article, I was struck by one passage which argues that precision medicine invested with social and environmental data could help deflate the power of crude biological race categories. As they write, with a growing depth of data, we have an opportunity to replace adjustments for race and ethnicity with more specific measures. In particular, race conflates a plethora of social, cultural, political, geographic, and biological factors together and can perpetuate systemic racism. Routine collection of social determinants of health in both research and clinical care in combination with more precise measures of environmental influences, habits, and genetic ancestry can provide more rational etiology-based adjustments and yield better risk stratifications and treatments. So there's so much to say there. It's pretty interesting. Look, as a gender sex scholar, I'm invested in pushing for the inclusion of social variables in biomedical research, right? I want to see the development of multidimensional biosocial variables. We're interested in gender studies in thinking about how these gender-related social variables interact with biology to produce the diversity and variation of gendered and sex conditions in the world. And I've written about this in all sorts of ways. As we argue in this piece in Cell Reports Medicine, why sex as a biological variable conflicts with precision medicine initiatives, we sort of use this as a wedge. Look, if you really mean precision medicine, we say, well, here's what you're going to have to do. And we kind of build this argument around the ethical considerations, around the deployment of sex as a variable in precision medicine, and argue, again, that there's more ambivalence in ethical weight to this. It isn't just all upside, including sex, and that in making these choices, there will be responsibility that needs to be attended to, and options that need to be considered. So we argue, actually, that a precision approach to biomedical research could bring a positive realignment of sex and gender science if, a la Denny and Collins, it shifts from blunt and uninterpretable sex categories to more of that contextual, variation-affirming kind of approach, integrated with the study of gender and sex-related environmental variables. So there are openings there to argue and for more contextualized approaches and to swerve perhaps away from these essentialist crude categories in these new data-intensive approaches in biomedicine. Of course, that's not where the current discourse is. The current discourse is sex difference research is precision medicine, right? And in recent years, women's health advocates have prolifically used the platform and discourse of precision to argue for sex as a biological variable. So there are the tensions and opportunities I see in this arena around sex as a biological variable, and thank you for the opportunity to speak with you all today.